Okay, lecture 4, we'll talk about affinity tag and fusion proteins. Affinity tag and fusion protein. So when you talk about different types of chromatography columns, we said the affinity chromatography is the most efficient chromatography methods because the binding that's happening between the target molecule and the resin is very specific. So there's no other protein, theoretically there's no other protein that can be bound to the column. So, you can capture the target molecule very efficiently. So affinity chromatography method is very efficient chromatography methods, right? Then the question is, so what if my protein doesn't have any ligand or any specific molecule that it can bind? And can I still use an affinity chromatography resin? Is there any way that will allow me to use affinity chromatography method? That's the question, right? So if that is the case, yes, the answer is yes. You can generate a tag to your protein molecule and then have the tag bind to its affinity column. So by engineering your protein to have a little tag on its tail, or maybe head or tail, and then you can have that tag bind to the resin, then you can use affinity chromatography. Yes, that's the answer. So that's what we're going to talk about here in this lecture. Okay, so if you look at this diagram, you can see if your protein is this blue part, I don't know what this color is, but if your protein is originally this part, but if you can somehow add an additional part to your protein, and if that additional part has very specific affinity binding with its target molecule, which is embedded on your resin, yes, you can use that column chromatography method to purify your protein. Okay, the unwanted protein, in this case the red proteins, will be purified away, so removed from your target protein. Only your target protein can be recovered, purified and recovered, right? So that's what you're talking about. Then you may wonder, so how do I make this tag attached to my protein, right? So that's where the genetic engineering will come into play. Okay, so again, so if you can clone the gene of your interest, so within a plasmid DNA that contains maybe your gene of interest, let's say this is your gene of interest, and then at the right tail of it or head of it, you put an extra tag expressing gene. So by combining your protein with additional tag protein gene, we call that a fusion protein. Fusion. Two different protein coding genes are fused together. That's what fusion protein is. Okay, so you create a gene that codes for a fusion protein. Okay, once you do that successfully, yeah, that plasmid DNA can be transformed into a host cell, and the host cell can make your protein for you. So in this case, maybe this is your protein, and then there's a little tag attached to it, right? And then the tag has affinity to target molecule. So now there's a column resin, okay, that has special maybe ligand or something stuck in them. Now your protein can bind to the column resin, and then you can purify it, so you can efficiently fish out your target molecule. Once that's done, it is just your protein only. That's what you need, not the tag part, right? So if we can genetically engineer a protease enzyme cutting site between your protein and the tag protein, yes, by adding a protease enzyme, you can cut them off, and then you can also further purify them, because there may be a big difference in their sizes. So you can use a size excursion chromatography to purify your protein out. So that is done a lot, actually, right? So here's another example. So your target protein right here, so it's fused with another tag protein. In this case, GST is a tag protein. GST stands for glutathione S transferase enzyme. It's a small protein, okay, that's attached to your protein. So that GST protein has very strong affinity with glutathione, which can be embedded on your resin. Okay, then your protein that's fused with GST can be purified over this column. Okay, so the other protein will be removed, and then once you have your protein stuck in the resin, and then you can elute your fusion protein by adding free glutathione. By computation, you can recover your protein. Same thing, right? So it's straightforward, very smart way of purifying your protein. So again, understand what fusion proteins are. Fusion proteins have extra tag attached to your target protein. How is it done? You can do it at the DNA level by cloning the gene expressing your protein next to another gene that expresses the tag protein. Okay, that is how it's done, genetically engineered, right? So the tag can be added on the N terminus of your protein or C terminus of your protein, whichever is more stable and better in terms of purification. Okay, so that's how it's done. Tag is added. So example, in this plasmid DNA, so GFP is a green fluorescent protein. Yeah, you can use that as a tag because this is a strongly hydrophobic protein. Maybe you can also, there are some antibodies that are binding with GFP as well, so you can use antibody column to purify your protein once it's tagged, your protein is tagged with GFP. So again, GFP coding gene and then your protein coding gene is cloned together side by side. In this case, if it's the 5' and 3' end of your gene, so GFP is added to the N terminus of your protein. Okay, so P is a promoter sequence, T is a terminator sequence. So transcription will take place from here to here, so everything will be transcribed together, and when that mRNA is translated, here you go, you have the fusion protein, GFP plus your protein. So now you can purify this protein over antibody column. Makes sense? So that's done a lot in the maybe GLP scale when you study your protein and try to purify it. Okay, so the tag can provide very strong benefit to a recombinant protein, because the recombinant protein, the fusion protein, can be purified. So this is probably a, I don't know, Comasi staining or silver staining. So uninduced and induced, there's some kind of expression system that allows a strong induction of your gene expression. Seems like that is a target protein before and after the gene expression induction. Very small level, much higher level, right? And then as you can see, when you have your protein expressed, there are maybe hundreds or thousands of cellular proteins that are all expressed together. And the question is, how can you purify that, okay, into this single protein? So if you can run it over chromatography system, you can purify it, maybe affinity chromatography of some kind. So if it's a GST only, yes, you can purify it over glutathione column, okay? But if it is tagged with something, so originally GST only is about 27 kilodalton, right? But if that is fused together with your probably protein, in this case GST is fused with myosin 1 protein, so it's GST plus myosin 1 protein, which has a size about 70 kilodalton or something like that. Okay, uninduced, induced, there is that protein fusion protein, and then you will probably purify it. Okay, so that's the protein, nothing else. What are these then? Those may be the isomer, probably protease degraded protein. If this is chopped up into small pieces, probably that's what they are. Okay, so here you go, the same thing. So it shows you how efficient it is to purify your protein. Okay, this may be on Western blot analysis, I think. Okay, we'll talk about Western blot analysis later on. So understand the concept of, tagging your protein at the DNA level, okay, and then how that helps you to purify your fusion protein over a column. Okay, once your fusion protein is purified, and then you can just cut up the tag, just recover your protein only. Okay, understand that concept. So another example, GST tag we already talked about, right? It has a strong affinity against the glutathione. Okay, so you can use the glutathione column to purify it. And what is that? There's another tag called 6-histidine tag. Okay, made up with 6-histidine, 6 amino acid, and that has a strong affinity against the nickel ion. So you can purify the fusion protein, your protein, and the 6-histidine tag, and then you can run it over nickel ion, then you can purify it very efficiently. Okay, so you can create several different fusion proteins. Among those, let's talk about GST fusion protein. So you're going to add a tag called GST. GST stands for, again, glutathione S transferase. It's an enzyme that can transfer a thiol group from one protein to another. So its substrate is thiol group. And then what is it transferring? It's transferring as S thiol groups. Okay, and it has a strong affinity against the glutathione. Okay, so one of the most commonly used tags, and it can be tagged on the N-terminus and C-terminus. Okay, so these enzymes exist in eukaryotic cells. There are a lot of these enzymes, GST enzymes. About 10% of cytosolic protein is made up with this enzyme, GST enzyme, in the mammalian cells. Okay, so again, if you have a resin containing glutathione, okay, your protein that's tagged with GST will bind, because GST binds with glutathione very specifically, right? So your fusion protein is binding, okay, and other proteins are washed away, and then you can elute your bound tag fusion protein by adding extra glutathione. So there's a competition between, this glutathione on the resin and a lot more glutathione coming in. So through the competition, your protein will be eluted. So the extra glutathione will bind in this case. Okay. So reduced glutathione looks like this, and oxidized glutathione, it can become a dimer, and it may look like this. Okay, then GST binds both. Okay, so binds the glutathione, then in the reduced condition, in the oxide, it can transfer them into this form. So GST isomer forms are normally found in bacterial cells. Thus, endogenous bacterial proteins don't compete, are normally not found. So GST is a eukaryotic protein enzyme, but it is not found from bacterial cells. That's what it means. So if your target protein is a prokaryotic protein, a prokaryotic cell protein, then it is good to use GST, because there's no GST-like enzymes found in bacterial cells. Okay, so there's an advantage of using GST tag for prokaryotic protein. Your target protein happens to be a prokaryotic protein, and you are trying to make it from prokaryotic cells. Yes, GST is your choice. Okay, we already talked about this diagram. Okay, so you can put it on the N-terminus or C-terminus. Okay, in this case, the N-terminus. Okay, so here you go, the tag on the N-terminus of the fusion protein. So there are column resins available with glutathiones, right? So same thing over and over. That's a tag, fusion tag and fusion protein, and then they are fished over glutathione column. Okay, you can purify it. So before purification, after the purification, that's a size marker, of course. That's a commercially available plasmid DNA that contains GST coding gene, and then you can clone your gene of interest after that. Then it's going to be N-terminus GST tag, right? So if you do a chromatography column, if you look at the chromatogram, it may look like that. So it's affinity chromatography chromatogram, because you can see multiple peaks combined together as a clump, and then there's a target protein coming out. Binding buffer PBS at pH is 7.3. Elution buffer TRIS, similar pH 8, but you add a bunch of glutathione that are reduced, then you can elute your target protein. Okay. And then the second tag that is used a lot is called 6-histidine tag. Hexa means 6. So if you clone a gene that codes for 6 histidines in a row, so 6 times codon made of 6 nucleotides, so about 18 nucleotide long, very short, you can make 6 histidines in a row, and that 6 histidine can form a unique structure like this. We call that an imidazole structure. It's a strong nickel binding structure can be made. So that will work as a tag. So I'm going to stop right here. I'm going to continue.